Hello everyone, greetings. Thanks for the invitation. It's an honor to present my talk in this conference, and the topic of my talk is essentially in situ study of corrosion dynamics. Next slide. First thing first, why in situ is important? In situ approach is important. Well, if you're provided with these two images, one before the sample is exposed to the environment and the other when it's taken out of the environment finally, what you expect from the in-situ approach normally, you have to make a lot of speculation about how this got to this final stage. So the understanding would be based on the speculation. On the contrary, if you are provided with this movie where you can follow the process dynamics in real time, this provides a much deeper understanding about the mechanism of the process. Right, so because you can follow things like where the process initiates, how it progresses with time, whether the process is homogeneous and heterogeneous across the surface, these sort of things. Next slide. Here you see a suite of complementary in situ approaches that we have developed in Cambridge. Why a suite of approach? Because one specific technique could be more efficient to unveil specific facet of the same phenomena. Well, for each of this technique, we have custom-made in situ cells using which we drive the experiment in situ, except for this liquid TM, which has its own commercially available cell. Using that cell, we can recreate conditions of industrial interest, and we can also execute spatially and temporally resolved mapping of the local conditions and species. This could be used for so many applications, including the corrosion of the battery interface, catalysis, degradation of catalyst, but today's focus will be on corrosion of steel in a CO2 saturated anoxic brine, which is also known as sweet corrosion environment. Going to the next slide. And because of the time constraint, I'm going to briefly talk about only these three topics highlighted with the red box. Next slide. Starting with the simplest first, optical microscopy coupled with confocal Raman spectroscopy. This is the complete setup to make the system completely anoxic. I'm not going into the detail of this setup and design. You can refer to this reference. This is how the cell look like, the in situ cell. You can see the sample, the electrode. So what you can do, you can see the progress of the process using the optical microscope, and also you can track the Raman spectroscopy at the same time in situ. So what we achieved using this, just to start with, we started with a metallographically ground and polished sample steel. This looks like that. And then we nitrile etched the sample. The purpose was to identify a specific domain. We know that for nitrile etching, the ferrite phase looks brighter and the pearlite phase looks darker. These are the corresponding SEM image in secondary electron and backscattered electron mode, ferrite and pearlite. When you zoom in, you can see that for the pearlite, alternating layers of cementite and ferrite, which makes it much more rougher in terms of texture. And this is how the ferrite phase looks like when you zoom in. And then, as we identified this specific domain, we reground and repolish the sample when it looks like this, and then exposed to the sweet corrosion environment. As you see, you can still identify the particle, the domain, from its shape. This is perlite and this is ferrite. You can see the contrast inversion. This looks brighter and this looks relatively darker, and in it hidden is the mechanism of the sweet corrosion process. Anyway, when you look in the SEM images for this in secondary electron, and the backscattered mode looks like this, and when you zoom in, you can see the perlite phase looks much smoother compared to the ferrite, and that's why it looks brighter. We'll explain it later a bit more. Next slide. And then we followed the growth kinetics of the corrosion product that forms on the surface. In the sweet corrosion environment, we see only one phase, which is iron carbonate, iron 2 plus carbonate or siderite. We saw the growth kinetics of that siderite product in perlite and ferrite. We see the growth rate of siderite in perlite is much more faster compared to the ferrite phase. And using these two facts, we modeled a mechanism for the sweet corrosion process of steel in the early stage. This is the schematic of the steel in a sweet solution environment. To start off, we can assume that the carbonate and bicarbonate ions are distributed uniformly throughout the solution, and this is the perlite phase indicated with alternating cementite and ferrite layers. Cementite is cathodic, ferrite is anodic, so you can imagine inside the perlite phase there will be numerous microgalvanic couple which will drive the dissolution of this ferrite phase much faster compared to this pure ferrite phase. And when when the iron starts to dissolve from this ferrite layer, there are numerous microwell forms inside the perlite phase, which makes the movement of this ion transport of this ion much restricted, and which causes the supersaturation of the siderite phase to reach much quicker, and it precipitates and fills up this gap, the microwells, and that's why it looks much smoother. On the contrary, in the pure ferrite phase, ionic transport is not restricted, so it goes on dissolving until it reaches to the state of supersaturation, and that's why it looks much rougher. And that's the reason of the contrast inversion. Because of this is smooth, this looks brighter, perlite phase. Ferrite is rougher, that's why it looks much darker, because of the scattering of the light. Going to the next technique quickly. So in-situ liquid TM, as I told, it has its own commercially available cell, in-situ cell from the Hummingbird. I'm not getting into the detail of the setup and the process. You can see this a reference provided here. We used the silicon nitride chip, which thickness at the viewing window is approximately 10 nanometer, and we deposited a layer of iron by E-beam of 50 nanometer and then annealed to make sure that is adherent to the film membrane. Going to the what we achieved by using this, we saw that the grain boundary is the preferential site where the nucleation occurs of the corrosion. You see, this is the movie, this is the breakdown. So it starts at the grain boundary and progresses along the grain boundary and then branches to the boundary of the next grain. This is an important finding. Going to the next technique, which is simultaneous small angles and wide angle X-ray scattering using high brilliance synchrotron X-ray radiation. And this is the setup, which is detailed. You can get from this reference here. This has been used in the I-22 beamline of diamond light source, and the reason for using this sax and wax simultaneously, this is used in reciprocal space, so the small angle sees the larger object while the wider angle sees the smaller object. As soon as any new species forms in the solution, sax should be able to find it out due to the change in its intensity. It will indicate that, and wax will tell you if that is crystalline or not. So the main purpose of using this simultaneously is to understand the nature of the species that is formed. Yes, and this is how the cell look like from inside. This is the foil, iron foil, as a working electrode, which is 10 micron thick, transparent to the X-ray. This is the platinum foil as counter electrode, and the reference electrode sits here. And this is showing you the scan grid across the sample solution interface. It will be a raster type scanning, and 1 pixel takes 5 seconds. The entire scan will take like half an hour, and then you can repeat the scan as many times as you want. What we achieved from here. Next slide. Here you see the wax pattern for the iron foil. Iron is crystalline, so it is going to give you wax pattern. This is the iron foil. The dark region adjacent to that is the pure solution region, which is uniform. So in the beginning there was no sax or no wax from here. As the new species starts to form, then it will be appearing. So this is the edge of the sample. You can see with time it starts to the iron starts to dissolve with time, and you can see the shift in the edge due to the dissolution of the iron. And here you see the wax pattern for the siderite and the sax. As you see, in the beginning we don't see any wax for the siderite on the iron foil, but we see some sax here, obvious, within first 1 hour. So which in and of itself is saying there is something forming which is not crystalline. But somebody may argue that this sax could be appearing because of the roughness, growing roughness, because of the dissolution of the iron, which is not true. We have proved that in this paper, not going into detail of that, but to avoid that confusion, we chose a layer which is in the solution, purely in the solution, adjacent to the foil but not on the foil. So this is purely in the solution, and we have shown that even here also the Sachs intensity is increasing from the very beginning within the fast hour gradually, but wax has been observed only after 355 minutes. So clearly there is something forming initially which is not crystalline, which is amorphous, and then that is transformed into crystal. The key thing to understand is that Sachs is intrinsically broader and weaker, while wax is localized and sharper. Seeing significant change in Sachs before the appearance of wax diffraction peaks is a strong evidence in and of itself that an amorphous product initially nucleates which crystallizes later. Past that, I'm going to the concluding slide now. So employing our suite of in-situ approaches, we have unveiled a number of crucial aspects about the dynamics of the sweet corrosion process. One of them is contrast inversion, and that is due to the inhomogeneous process kinetics in ferrite and perlite. And from our liquid TM, we showed that the corrosion process initiates at the grain boundary preferentially, and this information could be used for planning to stop and inhibit the corrosion process in a significantly more economic way, as opposed to inhibit the entire surface. And from our Saxon wax experiment, we have shown that the precursor formed is actually amorphous, and then it eventually transformed into a crystalline iron carbonate. And these are my actually colleagues in Cambridge, and this is my collaborator in the University of Manchester, and this is the diamond light source. And thanks for listening.